All right, this is Chapter 9, Part 2, Microbial Genetics. We're going to talk about how it is that we get genetic variation in microbial communities. I told you in the Part 1 lecture that DNA polymerase is very good at making nearly identical daughter cells when DNA is replicated. So, how is it that we get variation in microbial communities? How do antibiotic resistant strains arise? And how are these resistant traits transferred readily to other bacteria? So, bacteria have two ways in which they can adjust to changing conditions. They can regulate their gene expression, but they can also have genetic change occur. Genetic change alters the genotype of an organism, which in turn may cause a change in their phenotype or observable characteristics or express traits. Genetic change can occur by two mechanisms, either mutation, so a change to the DNA in the chromosome, or gene transfer. So a mutation is a change in the existing nucleotide of a cell's DNA, and it's passed on to daughter cells. So it's part of that bacterial chromosome. Passed on to daughter cells, and the modified organism would be called a mutant. Right? And by the way, we're all mutants. Every single one of us has DNA that's present in our chromosomes that wasn't there in our parents. It's usually only happening in traits, parts of your chromosomes that don't code for things, but sometimes they do. All right, so let's look at some examples here. Since mutation arises in a single cell and then passes it on to the next generation, that's called vertical gene transfer. So we have the original cell, a spontaneous mutation occurs, a mutation in the genome. So when the cell divided to produce two daughter cells, a change happened, an error in one of them that wasn't caught by DNA polymerase. When that cell goes to divide, its daughter cells also inherit the mutation. And when those cells divide, their daughter cells inherit the mutation. This is vertical gene transfer. It's passed down through generations. Spontaneous mutations occur in a cell's natural environment. So random events can happen. 1 in 10 billion base pairs are different during DNA replication. What enzyme makes the mistake? Oops, gone too far. So what enzyme is responsible for base pairing? Good, DNA polymerase. So DNA polymerase is very good, but it's not perfect. So 1 in every 10 billion letters, it'll get wrong. And that causes a mutation. There are different types of mutations. So there could be point mutations, a substitution, so when one letter is swapped out for another, an insertion, when a new letter is added into the sequence, or a deletion, where a base pair is omitted. And these last two, insertions and deletions, cause frame shifts. Remember that when DNA is transcribed into mRNA, it's read as 3-letter codons when translation happens. And those mRNA codons are used to gather amino acids and assemble them in order. If you're reading in groups of 3, but one gets deleted, then all the letters in that sequence shift over by 1 if one's added or deleted. And it can affect all the amino acids, right, that come downstream from that. Whereas a substitution generally affects just a single codon. So suppose the DNA code is represented by these letters. We group them into triplets like codons. All right, the substitution of a single letter or nucleotide that could change the meaning of the sentence or result in a meaningless phrase, right? Like if this C is replaced with an R, then it changes the meaning of the sentence. And sometimes if mutations happen, they might make nonsense things that don't make any sense at all. Similarly, you could have mutations that change the amino acid or that code for something that doesn't make any sense, like a stop codon or a start codon in the middle of the gene. Insertions or deletions are more serious. These are the frame shift mutations, and they affect proteins much more seriously. So we've got our original sequence here, the cat ate elk. If there's an insertion of an R here, all of a sudden, none of these amino acids make any sense, right? Similarly, if we delete one, nothing makes sense anymore. So these will not produce a protein that functions the way that the original gene sequences should have. All right, and the effects of mutations on proteins, they can be silent. Sometimes they don't make any difference, right? Remember when we looked at that codon chart, there's often more than one amino acid that codes for this, more than one codon that codes for the same amino acid. So they could be silent. They could not make any sense at all. They could be missense mutations or nonsense mutations. So a silent mutation is a change in the DNA that results in no change in the amino acid sequence. Because of the redundancy of the genetic code. So if we look down here, if AAA is changed to AAG in the DNA, the mRNA codons are UUU or UUC, but both codons code for phenylalanine. So there'd be no change in the protein or the phenotype. A missense mutation is a change in the DNA resulting in a change in the mRNA that does cause a change in the amino acid. And the effect on the protein depends on the location of the protein sequence. If it's in a critical location, such as an active site of an enzyme, there could be loss of function. In a less important location, there might not be any adverse effects. A nonsense mutation is a change in the DNA resulting in a change in the mRNA that causes something like a stop codon. This results in a non-functional protein. You will need to know the different types of mutations. All right, so let's talk about their effects of mutations on proteins. So we have our DNA template, which is transcribed into mRNA. That mRNA is broken down into 3-letter codons, which are translated into amino acids, right? If there's a substitution mutation, then a base pair is changed, and that changes an amino acid. So this would be a missense. The new codon results in a different amino acid. Serine after the mutation, whereas the original coded for glycine. All right, and in this one, it's a nonsense. A new codon is a stop, resulting in a shortened protein. So instead of having a whole string of amino acids and then a stop, there's a stop codon that results too soon and that never makes a protein that functions. All right, so those were things for change in the chromosome. But how are resistance traits transferred readily to other bacteria? Gene transfers occur when a gene is acquired from another non-related organism. So not by cell division, but some other mechanism. So horizontal gene transfer, we see here's a cell. It's got chromosomal DNA, but it also has this extra plasmid. The plasmids can be transferred from one cell into another. And then once a plasmid is inside a cell, it can be passed on by vertical gene transfer as the cells divide. So let's look more into how this plasmid can transfer from one cell to another if the cells themselves aren't dividing. So gene transfer results in genetic variation, and genetic variation is critical for survival. If all bacteria were identical, they could be easily wiped out by the same thing, right? So there's 3 ways genetic transfer can occur in bacteria: transformation, conjugation, and transduction. These methods are responsible for the spread of antibiotic resistance genes. So, transformation is the uptake of naked or cell-free DNA by a competent cell. So, that DNA might just end up free-floating in the environment if the cell is lysed in a population of bacteria. And some genera of bacteria secrete segments of DNA. So one way or another, bacterial DNA can just be floating around in the environment. It makes it through the cell membrane and becomes incorporated in chromosomal DNA. This is called transformation. This is a very famous experiment conducted by Frederick Griffith in 1928. He had 2 strains of Streptococcus pneumoniae. One was a virulent strain, a strain that causes disease, and he'd also noticed these bacteria had a capsule. They cause pneumonia. He also had an avirulent strain that he observed has no capsule and causes no disease. So, for his first experiment, Griffith had living encapsulated bacteria. He injected it into a healthy mouse. The mouse died, and he cultured the bacteria from the mouse. Colonies of encapsulated bacteria were isolated from the dead mouse. Next, he takes living non-encapsulated bacteria and injects it into a healthy mouse. The mouse remained healthy. He cultured bacteria from the healthy mouse, which he'd injected, and colonies of non-encapsulated bacteria were isolated from the mouse. Phagocytes had controlled bacterial proliferation. So there were bacteria, but they weren't taking over and causing problems. In the third one, he heat-killed encapsulated bacteria and injected it into the mouse. The mouse remained healthy, and there were no colonies isolated from the mouse. Right? So all of this is pretty much as you'd expect to see. No major surprises. Until now. Living, non-encapsulated, and heat-killed encapsulated bacteria. So neither one of those 2 things killed the mouse on their own. But he mixed the 2 non-lethal things together, injected them into the mouse, and the mouse died. When he cultured the bacteria from the dead mouse, he found colonies of encapsulated bacteria were isolated from the dead mouse. So what happened? Well, the bacteria were transformed. DNA from the heat-killed virulent strain was free-floating in the environment. The non-pathogenic strain of bacteria was able to uptake this DNA and incorporate it into its own genome. The recombined chromosome you can see down here, where it has that B gene here for pathogenicity, and the transformed bacteria are now pathogenic and encapsulated. You should review this Griffith's experiment, maybe draw sketches or practice drawing it out so that you understand the steps and its importance. All right, another way that genetic change can occur is through conjugation. When one bacterium passes some DNA in a plasmid to another bacterium. This involves plasmid transfer or sometimes short pieces of chromosomal DNA. And conjugation, as the name implies, always includes this structure, a sex pilus, a structure going from one bacterial cell to another bacterial cell, and genetic information is transferred through the sex pilus from one cell to the other. All right, the third mechanism for bacterial genetic change is transduction. Bacterial DNA is transferred from one bacterium to another by a virus. Now here you see a picture of a bacteriophage. This is a virus that only infects bacteria. How do multiple resistant strains arise and evolve? How are resistance traits transferred readily to other bacteria? And why must bacteria rely on these methods for adaptation and survival? These are some questions I want you to ponder. We'll discuss them in lab. Become prepared with your thoughts and questions about these, okay? All right.